Welcome to lesson 6.5. In this lesson, we will cover the other food sources that soil protists use besides bacteria, which we've covered in the last lesson. So in this lesson, we'll look at specifically at mycophagy or mycophagy, and this is where protists or any other organisms consume fungi. So mycophagy is the consumption of fungi, and nematophagy or nematophagy is where an organism, such as a soil protist, is able to consume nematodes. So that's what our lesson is going to be about these two groups of organisms that soil protists feed on, fungi and nematodes. All right, let's begin lesson 6.5. So in the last lesson, we've covered how soil protists feed on bacteria and the significance of that feeding behavior. But soil protists actually have many different types of food preferences. Those that feed by phagocytosis, or that are phagotrophic, meaning that they engulf particles, including live organisms such as bacteria, can also engulf other protists. So for example, several ciliates and some testate amoebae prey on other protozoa or other protists. And around 50% of 250 soil ciliates that were studied contained protozoa or other protists in their food vacuoles. Leptomyxa reticulata is thought to be mycophagous, so that means it consumes fungi, and we'll cover the topic of mycophagy or protists feeding on fungi. But these guys also ingest small amoebae. The giant mycophagous amoeba, Arachnola impatiens, consumes flagellates and nematodes because it is giant, so it can actually consume a whole nematode. And remember, a nematode is a multicellular organism, an animal actually. So these mycophagous amoebae actually also consume animals, which we'll talk about that later as well. The giant amoeba Vimpirella thorax consumes small diatoms, flagellates, and digests them in the digestive cysts that it contains. And it actually has been found to have reduced growth on bacteria and conidia, which are spores of some fungi. So this particular species actually prefers to feed on diatoms and flagellates. Some soil protists are detritivores, so that means that they consume humus or organic matter particles in the soil. And many testate amoebae are detritivores. Some naked amoebae also contain cellulase and chitinase enzymes, so that means that they are able to decompose organic matter, and even that organic matter which contains chitin, such as insect leftovers. The exoskeleton of insects contains chitin, and so do fungi. So there are various ways in which soil protists feed, and we can no longer accept them being only bacterivorous, as is often thought or often explained in soil food webs. And this is extremely important because all the different food sources that they can consume contributes greatly to a very varied nutrient cycle within the ecosystem as a result of these guys being present and being able to decompose and cycle nutrients that otherwise would not be able to be cycled unless these different feeding systems exist within the soil protist diversity. So it really shows how important it is to have a huge biodiversity of soil protists, as well as all the other organisms, but these guys are very important because they are at that initial stage of nutrient cycling, taking in, being so small in size. They are the first predatory organisms that begin the nutrient cycling within the soil food web. Very, very important organisms. Surprisingly, many soil protists are actually mycophagous. So mycophagy is the act of consuming or eating fungi, and some of the bacterivorous soil protists will consume yeast as well. Yeast cells are fungal cells. They're a little bit bigger normally than some bacteria, but they're a good food source for some of these bacterivores. Many unrelated amoebae feed on fungi. For example, the giant amoeba, which we have already mentioned earlier, Arachnola impatiens, consumes fungal mycelium and conidial spores, of the plant pathogenic Cochleobolus satitus. They remove the condylial wall, so from the spore, they remove a portion of the cell wall of the condylial spore, or some of the wall of the mycelium, and then digest the cytoplasm, so they can suck in almost the fungal cytoplasm from the fungal cells. There are 3 giant amoebae, including the genus Vampirella, that perforate fungal spore cell walls and or ingest the spores and digest them in special digestive cysts. Few amoebae are actually obligately mycophagous. That means that they can only survive on fungi and nothing else. They will not be able to survive consuming bacteria or other protozoa. So an example is Dermamoeba minor, which consumes hyphae and spores by perforating the cell wall and absorbing the cytoplasm. There are also mycophagous flagellates that eat nematophagous fungus, Drechmeria coniospora. Now, to clarify this, the flagellates eat the fungus and the fungus eats nematodes. So that's really interesting nutrient cycling. The nematode will actually leave a very carbon-based shell from its cuticle. It's the skin that it wears around itself, and it sheds that over time, but when it gets eaten, that cuticle is left behind. And that's being consumed by the fungus, and then flagellates consume the fungi. So that nutrient cycling, you can see it happening right there with the different nutrients being passed down from the nematode all the way to the flagellates, which are much, much, much smaller, but they're eating the fungi that consume the nematodes. There are also some obligate mycophagous ciliates. So once again, ciliates that will only eat fungi and cannot survive eating anything else. The image on the right comes from a very interesting research titled Interactions of Free-Living Amoebae with the rice fungal pathogen Rhizoctonia solani. And the reason for the research is because Rhizoctonia solani is a very prolific pathogen of various crops, including potatoes, rice, soybeans. It causes root rot and all kinds of other symptoms on plants. So this group of researchers decided to investigate whether different species of free-living amoebae can actually in some way control this pathogen. And what they've discovered is that the amoebae cause changes in the hyphae. They sort of become wrinkled and lose their plump look, like in this image here, and they sort of become all flaccid and wrinkled. So there's an effect of at least one of the species of the amoebae on the pathogenic fungi. And these arrows indicate whether amoebae have actually caused perforations in the fungi as well. These are scanning electron micrographs showing what happens when Vermamoebae vermiformis, so this is one of the species of amoebae used in this experiment, and Rhizoctonia solani were co-cultured, so being grown together. And what happens within those 24 hours of co-culture. And it turns out that the amoebae attach themselves and wrap around the hyphae of the Rhizoctonia, and this causes a shriveling of the hyphae that you can see here. They look different. So this image shows the varying intensities of the shriveling effect. And then when there was a separation of the two organisms by a membrane, after 24 hours, even though there was no direct contact between the amoebae and the hyphae, there was still a shriveling effect, as can be seen here. And then the remaining of the images show perforations that occur when the amoebae are co-cultured with the hyphae, so causing some sort of a dissolving effect on the hyphae. And really, this is just to show the possibilities of controlling pathogens using protists that are either antagonistic to pathogenic fungi or ones that can consume them. And it's not clear as to whether there was a consumption by the amoebae of the contents within the hyphae, whether they were actually feeding on the contents and hence the perforations to get the nutrients out, or whether it was just an antagonistic effect, an attack on the organism, and also some chemical interaction going on in order to cause this shriveling effect. But a very interesting study, and just to show those interactions that are happening within the soil and how beneficial it is to have protozoa such as amoebae in the soil because some of them can be antagonistic towards plant pathogens. So even though we don't know exactly what is going on, there's a definite effect, and this really is of interest to controlling this pathogen of the different crops by having these amoebae in the soil. So the idea of it would be to inoculate the soil with the amoebae when the pathogen's present in order to control the pathogen. So it turns out that mycophagy is so much more prevalent than it is given credit for. And in a review titled The Soil Food Web Revisited, Diverse and Widespread Mycophagus Soil Protists, there are some really key examples of how prevalent mycophagy actually is. For example, 2 flagellates, a testate amoeba and 4 genera of naked amoebae that were fed 4 yeast taxa, or 4 different taxonomic groups of yeasts, were able to grow and sustain themselves on this yeast food. So that means that these different species of soil protists can actually consume yeasts exclusively. And 4 of these studied organisms were able to thrive also on spores of plant pathogenic Fusarium. So once again, these organisms are actually feeding on fungi without the need to feed on bacteria. So it was concluded that mycophagy is common and plays a substantial ecological importance in terrestrial ecosystems, but is still very much so overlooked. So there needs to be an evaluation of those interactions between the different species of protists and their relationship to the fungi they use as food, which will by default influence the fungal community and change the functional trophic importance of the groups of organisms that both feed on the fungi and the fungi themselves. So this diagram comes from that review and exemplifies the food web, and we already have seen this several times where photosynthesis occurs, mycorrhizal fungi are attached to the plant produces exudates and organic matter, and those feed the soil food web ecosystem, particularly to start with. The mycorrhizal fungi exchange nutrients via the system, via the roots, and then the fungal decomposers and the bacteria feed both on the exudates and the organic matter. But the difference between this diagram of the soil food web and that of the usual or the common representation that has been given is that protists don't only just feed on bacteria, but also there are omnivorous protists, for example, that don't only feed on fungi and bacteria, but also on other protists, on the bacterial feeding protists, for example. And then there are also mycophagous protists, called mycophages in this case, in this diagram, and they feed directly on fungi, as we have thus far learned. And these protists, the mycophagous protists, also can feed the omnivorous protists. It is also very common that nematodes will also feed on protists as well, not just bacteria and fungi. And of course, arthropods are fed by the different groups of organisms, including the various protists. Some collembola, for example, which are springtails, depicted here, they actually feed on slime molds, which are protists, amoeboid protists, or even flagellate protists. So it becomes way more complex than is usually thought. And this is really important because, as I said earlier, all these relationships really determine how nutrients are cycled within the soil food web. And the more complex this soil food web is, the greater the biodiversity of organisms, and that translates to above ground biodiversity and supports more biodiversity in plant communities as well as a result. So it really ups the ante in terms of what's possible above ground if the soil food web is complex and entails the understanding that protists are a major part of the nutrient cycling and the interactions that they govern between plants and the associated soil food web network. So it is very important to keep unraveling the complexity of the soil food web, and also to ensure that we want to bring back that biodiversity to our soils and understand how it functions in order to desire to preserve it and give it its deserved recognition in plant nutrition. Besides consuming fungi, some soil protists actually consume nematodes, and this is called nematophagy. And examples of that are some large nematophagous amoebae. They can be from anywhere from 50 to hundreds of micrometers in size, and some of them belong to the vampirellid Arachnella impatiens species, but there are also others. And they actually contain digestive cysts within which they digest that nematode. So they are large enough to be able to engulf an entire nematode and then proceed with the digestive process in the digestive cysts. There are also nematophagous arcellanid testate amoebae, so these are called arcella, and we've discussed the arcella right at the beginning of the unit. And also some ciliates as well are able to consume nematodes. One very interesting experiment that was conducted ended up backfiring. This experiment was set up to discover how nematodes feed on amoebae, but what ended up happening was the opposite. The testate amoeba Cryptodiflugia operculata was observed pack hunting nematodes, and that included all the nematode species that were tested, including the active nematodes, dead nematodes, juveniles, and adults. So what was expected from the experiment was that the numbers of amoebae would drop with the introduction of nematodes, but the opposite happened. Nematode numbers started to drop and the amoebae numbers started to increase, and this is when it was noticed that it wasn't that the nematodes were consuming the amoebae, but the amoebae were actually pack hunting the much larger nematodes. So the way this happens is that one amoeba attaches the tail of a slow-moving nematode to the substrate, to the substratum, so something solid that the amoeba can attach the nematode to, and it's using its pseudopods to do that, and then it's probably releasing a chemical in order to attract other amoebae. And these completely immobilize the nematode, and then within 12 hours, the nematode is completely disintegrated, completely consumed by the group of amoebae that have pack hunted the poor creature. So these are some time-lapse photos showing this process from the beginning to the end, when the nematode is first attacked and then completely consumed by 12 hours. And you can read more about this quite fascinating accidental discovery in pack hunting by a common soil amoeba on nematodes. So these are some of the other relationships that are possible in the soil food web ecosystem. There are also other interactions, and that includes nematostatic activity, where amoebae actually produce exoproducts to repel nematodes. And the opposite can be true, where nematode exoproducts cause an increased encystation of amoebae and decrease their growth. So these are just competitive interactions that can occur. Also, some flagellates can consume nematodes when their numbers are really high, so when their abundance exceeds a particular threshold, and they are then able to consume nematodes. And when flagellate densities are lower, nematodes consume the flagellates. So it just really depends, and it's very specific and dependent on what's happening in that soil ecosystem as to how these interactions play out. And they are quite surprising, and they all play a significant role in nutrient cycling to the plant and in the maintenance and stability of the ecosystem. So it just really depends on what is happening within that particular soil as to how these potential interactions eventuate. But the most important thing to stress out is that, once again, biodiversity and ensuring that there's plant cover and moisture and the right conditions for the soil ecosystem to thrive, that these conditions are maintained in order for these interactions to have the most positive effect on plant growth and biodiversity increase within the ecosystem. So we have come to the end of lesson 6.5, and in our next lesson we will continue looking at soil protists and their role in nutrient cycling. We'll particularly focus on mixotrophs. So these are soil protists that have both photosynthetic capabilities and are able to hunt other organisms as well. We'll look at the fascinating world of slime molds, because they are soil protists that come together to form different formations that we can actually see with our naked eye. A macroscopic image of one you can see on the left here, an image by Sarah Lloyd, who has written the beautiful Australian book called Where the Slime Mold Creeps. And this is what some of these soil protists create. They create these beautiful structures, so we'll learn more about those. And we'll also cover microbiotic crusts, and this is where soil begins to form on desert sand, and how organisms come together to form these crusts, and how the different organisms we have so far spoken about can play a crucial role in the formation of those crusts. So, you'll be hearing from me again in lesson 6.6. Until then, have a good one. Bye.